Hello, everyone, and welcome to our third week of climatology. We are talking about paleoclimatology now, which simply means, sort of before current measurements. Paleo just meaning old or sort of pre-industrial type measurements. And so we're going to talk this first lecture about a lot of the ways that we have measurements from paleoclimates, so past, really, distant past climates. And then the next lecture, we'll spend some time talking about how you use those to sort of reference each other and piece together this really impressive climate lineage that we have going on. So first, fun fact for the day. I'm sure you guys have all seen, something like this, some sort of image like this or a map like this showing kind of what, the continents looked like in old times, right? In paleo times, or even, before paleo times, way back in the time of dinosaurs. So this is this is linked off of the book, right? This is one of the references that he points out as pretty cool to go check out. And so we've got this, the time period here is 25 million years ago. But what I like about the simulation is it talks about the amount of oxygen in the atmosphere, amount of carbon in the atmosphere, and interestingly, day length. It's not so interesting at this time scale, but if you go back, 150, 200 million years ago, the daylight or the day length is significantly shorter. But I like this because, fun fact, this is 327 parts per million for carbon dioxide, and look at how much ocean we had at that point. We would probably be underwater. It's really hard to say. We're somewhere in here, right? At least most of us. So we might be okay, but certainly Bandon and Brookings and sort of the most of the Oregon coast is underwater under this time period. In this time period, and then you fast forward a few years to 21,000, and we're right at the end of the ice age, right? This is when the glaciers were down throughout most of North America and just starting to recede. And it's already, some of it's receded out of our area, but you do see these Pleistocene era lakes happening. So that's Lake Lahontan, that's the Great Salt Lake, right? And Lake Lahontan actually is at some points much bigger. But this is just kind of a reminder, fun fact of the day. The Earth's been through some significant changes, right? Some really significant. And then you look at the parts per million up here, carbon dioxide, because everything was frozen, very low, right? Okay, so when we're talking about paleoclimatology, it's really important to establish proxies and understand what proxies are and how they're used. So I got a couple different definitions here. I mean, generally, it's just a proxy is a figure that can be used to represent the value of something in a calculation. And I like to think of like the gross domestic product, right? That's a single figure that can be used to represent something, and relatively complex, really. But it's sort of a synthesis of all kinds of other data, and in itself may not mean very much in specific contexts, but it's a nice relative number, right? A nice relative thing. So in the context of paleoclimatology, it's, proxies are basically data that are highly related to climate, but are not direct measure of climate. The USGS goes even further, and they say it's the physical, chemical, and biological materials preserved within the geologic record that can be analyzed or correlated with climate or environmental parameters in the modern world. And so basically, right, proxies are a way of taking measure or inferring measurements of climate from the past when we didn't have, weather stations or thermometers or even, sometimes humans to go out and measure these things, right? It's a way of inferring past climates from things that are preserved, either in the geologic record or in other ways. And so there's a lot of different paleoclimate proxies. This is just a sampling of them, and I'm going to talk about not all of them, but a few of these, just to give you an example of what these look like and what they mean and how they're analyzed and all that fun stuff. So, and I like this figure too. I kind of found this, and this is what you would typically find anytime you're talking about paleoclimate. People often reference these four different metrics often together, right? So you look at tree growth, and then you look at charcoal sediments in lakes, and you look at pollen, and you look at oxygen isotopes, and together you get a really comprehensive idea of past climates. I like this table too because I mean it talks about the source, but then it talks about the temporal and spatial resolution, which is really key, right? We were talking about this at the end of last week. Thinking about, well, what has high, either seasonal or annual temporal resolution. So either, spring versus or spring, summer versus fall and winter, or even just annual every year, versus some things have annual to decadal, and some things are multi-decadal to centennial. So every 100 years, or what is that resolution, right? So how much can you infer from a single sample? Can you infer multi-years, or does it need to be multi-decades? And then the spatial resolution as well, like how specific is it to the place that it's found? So tree rings are obviously specific to where that tree is, right? That's a really high spatial resolution. Charcoal pretty much too, because it doesn't get blown around like pollen, which is sort of moderate, or oxygen isotopes, which is like they're blown all over the place, and air mixes all the time, and so the spatial specificity of oxygen isotopes pretty low. um, so I like that. But let's talk about some of these actual specific proxies. So the first one I'm going to talk about is pollen, and this is a whole field of study called palynology, and it's actually something I did during my dissertation. We were required in my ecology, evolution, conservation, biology PhD to spend at least a semester studying with someone completely unrelated to your dissertation. So my dissertation was urban ecology. And so my research rotation, is what it was called, was actually spent with a guy named Scott Mensing, who's one of the world's leading palynologists. And so I got to spend a whole semester looking at pollen spores, and I actually still remember some of these. I think I'm pretty sure that's pine. I'm pretty sure that's oak, quercus, and I'm thinking one of these is a grass, and I just can't remember anymore. But anyway, we spent a lot of time kind of looking at pollen cores, and so pollen, right? You all know and probably have allergic reactions to them at some point during the season, right? Pollen's broadcast from all kinds of different plants and species, and a lot of it's wind blown and just gets blown all over Kingdom Come. And but usually some specific geography, and then most of it settles down on things, right? So it doesn't get intersected with the female parts so that you can actually reproduce. It just gets blown around and trapped, either in the dirt or in lakes. And the stuff that gets trapped in the lakes are what we're most interested in, because it can actually start to sort of start to settle. Gets into the water column, it slowly drops down, and it settles at the bottom. And this is key. And even for stuff that isn't wind blown, insects and other things, it all helps us understand what kind of vegetation may have existed at a certain time. And some pollen spores they found evidence of some of them up to 440 million years old, which is just a tremendous time period that we could potentially reconstruct based on pollen. And so the reason pollen is a good proxy for climate is because of basic ecological principles, right? Some things are going to grow only when the climate is favorable. You can't have pine trees in the desert. And so if you have a bunch of pine trees and all of a sudden they disappear for like 5 or 10 or 20 or 100,000 years, you know that the climate changed. And then when they come back, you know that it changed back. And so you look at this proportion of different pollen spores and species, and you start to kind of piece together what was happening in a given place. And so this is what it kind of looks like. This is someone going out and taking a pollen, or taking a sediment core from a lake. So we like to focus on lakes, and this works really well, especially for capturing post-Pleistocene. So the last 20,000 years of climate, because at the end of the Pleistocene, we had a ton of lakes, especially in North America and throughout Northern Hemisphere, that slowly dried up through the Holocene, that we're in now. Although a lot of people argue we're in the Anthropocene, which is different from the Holocene. But but so we have these lakes that are sort of remnant of these really large lakes, and so that means they've been fairly continuously wet. And so the Great Basin is a really awesome place to do this, because we know that most of those lakes have stayed wet since the Pleistocene, which means that they have a relatively continuous record of pollen accumulation in that area. And so you get out there on some catamaran kind of raft things, and you plop this big metal tube down, and you drive it down as far as you can, and then when you pull it up and you let the core dry out a little bit, you get something that looks like this. And look at all those striations. Look at those beautiful layers of sediment that have accumulated over time. And so you go in there and you find some, usually, little bits of charcoal or something with strong carbon signal, and you carbon date it. You use carbon 14, and you get a sense of, well, how old is the stuff that's at the bottom versus the top. So it's just an example from Battleground Lake in Washington. The bottom can be as old as the end of the Pleistocene, and the top is the present. And certainly you have different sedimentation rates based on all kinds of things, but you can get this general sense of where the time period that's represented by each of the level or layers in the sediment. So once you get something that's carbon dated, to kind of benchmark your core, you start looking at the abundance of things. And so this is how it's typically presented in a scientific paper type format. This is the depth of the core, so usually centimeters. And this is a small one, and this is not a very old one either, but they've carbon dated, and they've got it back to 1916 to 2006, right? And what you see is just that relative proportion of pine pollen compared to other pollen. So aquatic vegetation and some herbaceous kind of vegetation. Not sure what this concentration is. But you can see where there's high pine abundance, there's low herbs and that sort of thing, right? And when there's low pine, there's high herbaceous cover. And that's what they're looking for, is these big, strong signals. It's usually another famous one is sort of sedges versus grasses, because sedges have to have their feet wet, so they're a really good indicator of wetlands, versus grasses, are typically the thing that comes in after a lake or a wetland dries up, and that ratio between grasses and sedges is really interesting. So you always look at this relative proportion of things, which is kind of fun, right? And from that, you can kind of reconstruct then the relative climate that exists. You always base it with sedimentation rate as well. And this is pretty close to a lot of the other proxies, actually. So in the ocean, we looked at the planktonic foraminifera. Hopefully, I'm not butchering that too bad. This is well outside of my realm, but the same kinds of things get done, where you're basically looking for this little single-celled organism that lives in water and builds a chambered shell. And from that, the relative abundances of them, especially their shells, right, in a soil sediment core, indicates something about the conditions of the ocean or the water when that particular organism lived or died. So, and these go back to nearly 500 million years old, which is just crazy. So, certain species indicate certain water quality or temperature or salinity levels. It's really good for understanding ocean acidification because it's a calcium-based shell. So if it, thins or thickens, given decay and everything, regular decay, you can infer a lot about the ocean chemistry as well from these things. So they're a really, really popular proxy. And again, you're looking at the relative proportions based on the depth, which is then ground truth to some age. And then you take it all together and you say, oh yeah, at this point in the ocean, it was either warmer or saltier or drier or whatever. Another one is chironomids. So these are fun little midges that have been around for an awful long time, and that's kind of what it looks like. Pretty tiny little non-biting fly. This is their life cycle, and what's particularly key about this is that well, they're abundant as all get out. They're an invertebrate, so there's lots of them. But for some reason, their head in their larval stage preserves really, really well in the sediments of lakes. So anytime you're doing like a lake sediment for, palynology study, you're also looking at chironomids. You're also getting a sense of water chemistry and water pollution sort of metrics, because, like most invertebrates, they're highly sensitive to any kind of changes in sedimentation rates or influxes of new chemicals, certainly of warming and cooling and, fire and all kinds of things they're really sensitive to. So you get people that actually spend their, I don't want to say spend their life, but spend a significant amount of time looking for the heads of these wormy little larvae of this little midge fly. Because it turns out they just correlate super, super well to temperature and all kinds of other things. Pretty neat, huh? So just looking at ancient bug heads, you can start to reconstruct climate. Neat. So I'm sure everyone is super familiar with ice cores, and so I want to talk about that just a little bit. Obviously, that is a really, really awesome proxy for climate. Again, we're not measuring climate directly with the ice cores, but looking through the deposition of ice, we can tell an awful lot. So this is actually stuff and these guys are from the Desert Research Institute, where I worked for a long time, and they have an ice core lab. I worked next to the ice core refrigerators, freezers for many, many years, because it was in the basement, and that's where they stuffed grad students. So I got to know Larry, Curly, and Mo, the three freezers, with ice cores from Greenland very, very well. I walked by them, I don't know, 10 times a day for most of my grad school. So this is what they're doing. So they get these massive augers out there in really terrible conditions. They plop them out. They make sure that they're clean, right? They make sure they're well preserved. Because the last thing you want to do is spend all that money going out to Greenland, taking these cores, and realize that you've chosen the wrong place to drill, and there's some imperfection, there's some inconsistencies, or something happened, right? And then you take it to this guy. He was sort of the golden child at Desert Research Institute, Joe McConnell, right here. Still there, still very, very famous. He's got the ice core lab and the freezers, and he goes in there and he builds these fancy dancy machines here that will layer by layer take out, extract out and chemically analyze the gases that are trapped inside the ice core. And that's how we know the carbon dioxide levels, the oxygen levels, the ozone levels. I mean, so many particulates, so many of the atmospheric chemistry is trapped in these little air bubbles located in these cores. And I mean, this gives you a sense of, just like sediment deposition, we have ice deposition that grows and shrinks over time and depending on the surrounding climate. So you can infer also temperature and global climate systems from the width of these cores, or of each of these bands, right? These layers of information. But most importantly, you can get at that gas proportion, the ozone versus carbon dioxide versus oxygen and methane and all those things. And that's where we get our records going back 800,000 years. It was in the Greenland ice sheet. These cores, probably something that would be on the order of that long, gave us this long, incredibly long-term view of carbon dioxide in Earth's atmosphere. Pretty neat, huh? So we have the ice cores. Compare that then to, I should say tree cores, not ice cores, my apologies. Compare that to tree cores, which is a totally different temporal resolution. And hopefully some of you have played around with this a little bit in like ES 102 or something comparable. because you can chop down the whole tree if you need to, right? And get a nice cross section like this and get those really obvious annual rings where you have the summer and spring growth, where it's just growing the gangbusters, compared to the late summer growth where it's getting colder and resources are tighter, so it's not going to put on the girth as much. But you can reconstruct these annual cycles, right? Just chopping it down and getting a tree biscuit. Or you can core it, and I'm hoping that some of you have done that in like ES 102, where you can take an auger and you go up to a tree, and at breast height you stick it into the tree, and you pull out this plug that looks something like this, and from that you can start to count the amount of the rings. Well, the cool thing is when you combine all that together, you start looking at lots and lots of trees, and thin trees and thick trees and old trees and young trees, and you start to cross reference. So like say you got a core like this, and you say, oh man, the width of this, obviously within the same species, the width here matches the width here, and that happens to be the bottom of the core here. So we can kind of say something about the trees were, this is the same age between the two trees. And so if this goes back 1,000 years to 700 years, then we can say this probably starts somewhere around 800 years to 700 years, go up to maybe 500 years or whatever. And so the idea is that you kind of cross-reference all the way down your tree cores, until you've got a relatively complete record of tree growth, and therefore a proxy of climate. It's hard. I mean, it takes a lot of work to get chronologies that match up. If you core a tree or 10 trees and you just can't find any evidence of other trees experiencing that same growth pattern, you just kind of log them, and you say, or not like cut them down, log them, but you put it into a log and you say, well, maybe someday somebody will find something similar to this. But it's pretty challenging, but we've gotten to the point now where we've done this enough that we are able to cross-reference from one core to another, and get these really deep understandings of broad regional patterns driving tree growth. And in some cases, we actually have trees that just cover like the entire period we're interested in. And I don't know if you guys know about the bristlecone pine, but the bristlecone pine, this gnarly looking thing that lives in the Great Basin, White Mountains, California kind of stuff. I mean, it is the roughest, toughest. It's an ancient tree, like old leftover from the Pleistocene. I can't imagine it looked that scraggly back in the Pleistocene. But these things are remarkable. They live on the most harsh climates in the Great Basin, and they live forever. I mean, this is a core or a tree transect, I should say, of the trunk, and you can see just how far back it goes, 2,500 years. So that tree, when they cut it down, which is sad that they cut it down, was 2,500 years old. Right? It was there when Christ was walking on the earth. It was there when, the Egyptians were doing their thing. I mean, it's just a remarkable thing, like a tree, a single living organism would cover that. And so the oldest known bristlecone pine is actually over 6,000 years old. And that is in the Great Basin. So pretty remarkable. You can start to reconstruct these climatologies based on the width of these trees. I mean, it's really awesome then to take something like a bristlecone pine cut like this and compare it to pine and juniper all around the Great Basin, and you can always cross reference it back to here, right? You look for those similar patterns. So pretty neat stuff, huh? Last I want to talk about is plant macrofossils, and this is the fun stuff, I think. So plant macrofossils are simply plants that have been preserved in a big enough size that you could kind of see it without a microscope or doing any kind of extraction or whatnot. But I want to show you a couple. So there's certainly fossilized versions of this, which take millions of years to kind of fossilize. But then there's ones that aren't necessarily fossilized, and I want to just show you this quick video of one example of these preserved but not fossilized plant fossils. I mean, I don't know, plants, basically. So what you're looking at is the roots of sedges and grasses and other kind of wetlandy type vegetation. And what you see there is an ice lens. This is in the permafrost tunnel in Alaska. And what they found when they're digging, so this Army Corps, what they found when they were digging into it is that this wetland that formed this permafrost in particular froze very, very fast, so fast that it actually preserved the plant material, preserved the root structure. And I don't know if you saw it. I mean, everything, right? The soil biomass and everything is perfectly preserved in this sub-zero kind of refrigerator, right? And you can kind of see it here. I think maybe in the next little shot, you can see that there's still actually some, yeah, this one, some chlorophyll. Um, I mean, it's not alive, but it's green. It looks green, right? So even the artificial light we're showing on this still triggers a tiny bit of chlorophyll response. So you actually see some green right next to the ice, right? Isn't that amazing? And so it's really cool. I've posted this website here, and I want to do this quick show and tell on it because this is the website. And what it allows you to do is actually, this is the permafrost tunnel. So the Army Corps in the 60s said, hey, we want to see how far we can dig into permafrost. Literally, that was what they wanted to do. And so being that it was the 1960s and we were conquering everything else, why not, right? Like they invested a bunch of money into drilling. So they started drilling. That's the end of the tunnel. They started drilling into the permafrost. And they only got that far before they simply just couldn't do it anymore. They didn't have the energy or the bits or anything. It was just too hard at that point. So I can't remember exactly how long this is. It's probably 300, maybe 400 feet long in total. It's not super long. But what they've done now, so the Army Corps gave up on it. And it was really only an active thing for like 2 or 3 years, and then couldn't decide what they wanted to do. And so, luckily, a couple local scientists said, "Well, hey, there's like all these macrophytes and stuff in here. There's these cool plants and everything. Let's like use this as a research area." There's also, and I can't seem to get back to it, unfortunately. So you'll have to just do it on your own. That there's all these signs and everything, but there are like fossils um, of wood bison and other kind of animals from 14 to 20,000 years ago, like just in this sediment. So perfectly preserved, right? So it's pretty awesome, this resource that they have now. And so they've been just using it purely for research ever since. They put this door on it and to try to keep the cold in because obviously things are warming up out there, and that worked for about 10-15 years, but then they realized they actually have to refrigerate it, and it's more cost effective to refrigerate it than to decommission it or try to bury it up or whatever. And it is yielding a lot of good research. And so there's a big old refrigerator that keeps this cool now because otherwise this place would become melted. But you kind of see, you look up at the roof, and it was just amazing. You're literally walking like through a wetland that existed 50,000 years ago. So pretty neat stuff. I'll see if I can go in here. Yeah, you can kind of see the permafrost and the different lenses. The permafrost, that's how it kind of works and operates. But pretty neat. Oh yeah, that's 50 meters. So yeah, like I said, it's not very. They didn't get very deep at all. And these are thermocars, so basically melting kind of pool areas. And that's the end of it. Pretty remarkable, huh? So from this, you can extract out, plants and well, bones and animal tissue and that sort of thing. But plant tissue that's still actually active, and you can do DNA on it. You can understand, I mean, just so much about the climate, And again, this is most of those plant fossils in this case are going back about 20,000 years, maybe 25,000 years. So a remarkable resource for understanding past climate. So we have plant microfossils in that context, but my favorite, absolute favorite, is this. So Julio Betancourt, I never met him, but I worked with a lot of folks that worked with him. He's holding this thing called a midden. I wonder how many of you guys know what a midden is. Looks like a rock, doesn't it? It's kind of all gnarly and everything. It's kind of cool. But if you know what a midden is, you know that that is not a rock. It actually belongs to this character. And what that is an amberant. So pack rats, any kind of pack rats, bushy-tailed wood rats, all kinds of animals, these little mammals, they like to use the same den over and over and over again. And not just for like generations or hundreds of years. And sometimes, in some cases, it's like thousands or tens of thousands of years. The same little cave or little enclave place where they go and live is the same. And what they do, the reason people who are hoarders are called pack rats, is because they literally they grab stuff from the environment and they stick it into their home, their cave, their midden. And but what they do that's particularly useful for talking about paleoclimatology is that they do this for all tens of thousands of years that they use this. And you say, well, bug whip, it kind of decomposes and everything. Well, not exactly. What they do actually is pee on it. And they pee on it 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 for 27,000 years sometimes. And so in these amberants, you can actually have 27,000 years worth of plant material, or sometimes like archaeological, arrowheads, or like bird feathers, or all kinds of stuff, because these things say, oh, shiny, oh, food, oh, that's cool. I'm going to grab it, stuff it into my collection, and then I'm going to pee on it, and I'm going to forget about it. And then, for the next 10,000 years, my descendants are going to pee on it and forget about it. And so scientists started figuring this out in the mid 50s, I think, that these things are just gold mines for historic climate. Because again, you can radiocarbon date, certain material in it and get an idea of stratification of the materials that you find in these things. And here's a close up of it. I mean, you can kind of see the crystalline urine. It's kind of nasty. I worked across the hall from the guy who did this at Desert Research Institute, and man, when he was dissolving the urine, I gotta tell you, you don't want to be there. It's just some nasty stuff, but it's so amazing. Like you see wood fibers, you see rock. I mean, rock sometimes, but you see big chunks of wood that they've drug in. You see all kinds of stuff that is perfectly preserved in their pea nest. So you thought coprolites were cool. No, no, no. Amaranths are definitely pretty awesome. They just preserve so much. And so, like I said, archaeologists, paleontologists, all kinds of folks use these pack rat middens to understand just, and that's a pretty high spatial resolution, right? Pack rats aren't traveling a ton of distance. So it's a pretty specific like geography that then can be reconstructed based on what those little guys collected. So that just gives you a sample of some of the proxies that we use for understanding these really ancient climates. So like I said, in the next lecture, I'll get into, okay, how do we actually like derive data from this and start to compare it and reconstruct actual climatologies, those temperatures, those departures from normal and those kinds of things.